Reach up and touch your ear. Seriously, run your finger along the rim. That shape is not random. Before you were even 2 months old, 6 tiny pieces of tissue had to rise, meet, fuse, and fold into the structure beneath your fingers. The result is so individual that a camera may be able to recognize you from it, so precisely shaped that your brain uses it to tell whether a sound came from the ceiling or the floor. And so ancient that parts of its construction blueprint reach back to ancestors that lived underwater. Your ear may also contain a vestigial bump, expose a genetics myth you learned in school, and carry a tiny hole that leads to a tunnel beneath the skin. And the strangest part? Your nervous system may still be trying to move your ears, even when they refuse to move. Keep one hand nearby. By the end of this, that object on the side of your head will feel less like an ear. Start at the top and trace the curled outer rim and move about a third of the way down. Some of you will feel a small, firm bump, a tiny point interrupting the curve. It has a name: Darwin's tubercle. Darwin wrote about it in 1871, but the observation came from a sculptor named Thomas Woolner. While modeling human figures, Woolner kept noticing the same little projection on real ears. He told Darwin, and Darwin compared it with the more obvious pointed ears of other primates. That is why the bump is often described as a possible vestigial feature, a faint echo of an ear shape that was more pronounced in some of our distant ancestors. It is not a spare organ, it does not improve your hearing, and it does not mean you are more evolved or less evolved than the person beside you. Evolution is not a ladder, and nobody is standing on the top rung. Darwin's tubercle is simply one variation in the complicated process that folds a human ear. In other words, you are not carrying a defect, you are carrying a variation in the fold. Now slide your hand to the bottom, the soft, fleshy earlobe. Does it hang freely below the point where it meets your head? Or does it blend into the side of your face in a smooth curve? If you took a basic biology class, you may have been told that this is controlled by a single gene. Detached is dominant, attached is recessive. Draw the square, fill in the letters, predict the baby. Beautifully simple, but also wrong. A large genetic study found dozens of regions of the genome associated with earlobe attachment. And real earlobes do not politely sort themselves into 2 boxes. They form a spectrum: fully free, partly attached, strongly attached, and everything between. Your earlobe is not a coin flip, it is a construction record. Early in embryonic development, 6 small swellings called the hillocks of His appear around the region that will become the ear canal. They gradually merge and reshape into the outer ear. And sometimes the final structure varies, even between close relatives. Look at the ears in your family and you may see the same ingredients assembled in slightly different ways. Same family, similar instructions, but different build. Those differences are also why ears interest people far outside biology. Fingerprints get most of the attention, but the geometry of an ear can also act as a biometric. The rim, inner bowl, folds, angles, and lobe create a pattern that computer vision systems can measure. An ear is not universally better than a fingerprint or a face, but it has one strange advantage. Cameras often see it when they cannot see much else. A mask or sunglasses can hide much of the face. Almost nobody remembers to disguise their ears. Researchers have tested ear recognition systems for security and identification, with performance depending heavily on image quality, angle, hair, and the particular system. Even identical twins can have distinguishable ear shapes because development is influenced not only by shared DNA but also by tiny differences during growth. Genes write the instructions. Development performs them live, with no rehearsal and no undo button. But not every interesting mark on an ear is present at birth. Look at the earlobe again. Some people develop a distinct diagonal crease running down and backward across it, often at roughly a 45-degree angle. It is called Frank's sign, after physician Sanders T. Frank, who reported the observation in 1973 after noticing it in patients with chest pain. For more than 50 years, researchers have argued about what it means. Numerous studies have found that the crease appears more frequently in people with coronary artery disease. Other research has linked it with stroke risk and markers of disease in small blood vessels. One proposed explanation is that the earlobe contains soft tissue supplied by tiny blood vessels. Changes affecting small vessels and connective tissue throughout the body might become visible there as a crease. It is an unsettling idea, a process hidden inside the body leaving a mark where anyone could see it. But here is the responsible version, not the viral version. Frank's sign is not a diagnosis. It becomes more common with age, and so does cardiovascular disease. Healthy people can have the crease, people with serious heart disease may not. A doctor cannot read your medical future from your ear. At most, it is a clue that has to be interpreted alongside actual risk factors and proper medical tests. Your ear can suggest a question. It cannot deliver the verdict. Now move your finger forward to the place where the top of the ear meets the temple. A small percentage of people have a tiny opening there. It may look like an oversized pore or a pinprick dimple. It is called a preauricular pit or preauricular sinus, and beneath the surface, it may continue into a short passage or a branching tunnel. Its prevalence varies widely between populations and is generally higher in parts of Africa and Asia than in Europe. Remember the pieces of embryonic tissue that build the outer ear? A preauricular pit is thought to result when tissue in this region does not join completely during development, and this is where the story becomes ancient. The human head and neck develop using structures called pharyngeal arches. In fish, related embryonic structures contribute to the apparatus supporting the gills. In humans, they are remodeled into parts of the jaw, face, throat, and ear. A preauricular pit is not a leftover gill. You never had functioning gills. The similarity is deeper and more interesting than that. Evolution did not throw away the old construction system and begin again. It renovated. The same ancient developmental toolkit was modified, redirected, and reused. A shark uses versions of those instructions to build structures associated with breathing underwater. Your embryo uses related instructions to help build the side of your face. The blueprint was not replaced, it was edited, and you are wearing the edit. Now try to wiggle your ears. Some of you can. Most of you just raised your eyebrows, tightened your scalp, and made the expression of someone attempting telepathy. Around each ear are 3 auricular muscles, one in front, one above, and one behind. In many mammals, these muscles rotate the ears towards sound. A cat can aim its ears like radar dishes. A deer can monitor what is behind it while its eyes remain fixed forward. Humans largely lost that ability as our evolutionary history took us in a different direction, but the muscles and parts of their neural control remain. Recent experiments measuring electrical activity found that these muscles can activate when people strain to follow difficult sounds, especially the muscle above the ear. The movement may be tiny or invisible, but the old system still tries. You have a reflex looking for a job. Somewhere between the sound and the brain, an ancient command whispers, "Aim the ear." The signal fires, but the hardware barely responds. So, if our ears do not swivel effectively, why are they covered in all those ridges and valleys? Because the outer ear is not merely a funnel, it is a physical sound processor. Sound from above strikes those folds differently from sound below. Certain frequencies are amplified, others are reduced. Reflections arrive with microscopic differences depending on the direction of the sound. Your brain learns to decode this altered pattern. For a sound directly above or below, the 2 ears may receive very similar left-right timing information. The folds provide extra clues, allowing the brain to estimate height and distinguish front from back. And the code depends on your particular anatomy. During childhood, your brain learned the acoustic fingerprints created by your own ridges, bowl, and canal. Experiments that temporarily alter the ear's shape can confuse those cues. At first, locating sounds becomes harder, but eventually, the brain recalibrates. Your ear does not simply collect sound. It signs it before delivery. Every noise arrives at the brain stamped with the shape of the person who heard it, and that shape does not remain perfectly frozen. You have probably heard that ears never stop growing. The reality is more interesting. They do not keep growing like a child's bones, but measurements show that ears often lengthen throughout adulthood. Cartilage and connective tissue change. Skin loses elasticity. Gravity keeps collecting its payment. The lobe can stretch, and the ear gradually appears longer. Some studies have measured average increases of roughly a fraction of a millimeter per year, although the rate varies between people and populations. So the ear you touched at the beginning is neither a permanent label nor a literal fossil, it is a living record. Its foundation was assembled before you were born. Its variations came from genes, development, and chance. Its muscles preserve an old command. Its folds transform sound into a private code your brain had to learn, and its proportions continue changing as the years pass. Your ear contains no single dramatic secret. It contains something better. Layers. An ancient developmental toolkit. A one-person acoustic filter. A family resemblance that refuses to follow a simple classroom diagram. A structure so ordinary that you've seen it every day, and so complicated that dozens of genetic influences, millions of neural calculations, and hundreds of millions of years of evolution are hiding inside it. You have been walking around with a biological autobiography on the side of your head. Go find a mirror, but this time, really look.